Pentose phosphate pathway, a pathway in which no ATP is consumed and none is produced. This pathway is also known as hexose monophosphate shunt. This pathway is required for generation of NADPH, and it also generates 5-carbon ribose sugar, that is ribose 5-phosphate, which is required for nucleic acid production. The pathway can be considered to have 3 stages. First is oxidative reaction, which yields NADPH and ribulose 5-phosphate. Only first 3 reactions of the pentose phosphate pathway are involved in NADPH production. Glucose 6-phosphate dehydrogenase catalyzes net transfer of hydride ion to NADP plus from first carbon of glucose 6-phosphate to form 6-phosphogluconolactone. The enzyme is specific for NADP plus and is strongly inhibited by NADPH. 6-phosphogluconolactonase increases the rate of hydrolysis of 6-phosphogluconolactone to 6-phosphogluconate. 6-phosphogluconate dehydrogenase catalyzes the oxidative decarboxylation of 6-phosphogluconate to ribulose 5-phosphate and carbon dioxide. Formation of ribulose 5-phosphate completes the oxidative portion of the pentose phosphate pathway. It generates the 2 molecules of NADPH for each molecule of glucose 6-phosphate that enters the pathway. The second stage is isomerization and epimerization of ribulose 5-phosphate. In this, ribulose 5-phosphate is converted to ribose 5-phosphate by ribulose 5-phosphate isomerase or to xylulose 5-phosphate by ribulose 5-phosphate epimerase. The relative amount of ribose 5-phosphate and xylulose 5-phosphate produced by ribulose 5-phosphate depends on the need of the cell. Example, ribose 5-phosphate is an essential precursor in the biosynthesis of nucleotides. So, ribulose 5-phosphate production is relatively high in rapidly dividing cells, in which the rate of DNA synthesis is increased. If pathway is being used solely for NADPH production, xylulose 5-phosphate and ribose 5-phosphate are produced in a 2:1 ratio for conversion of glycolytic intermediates in the third stage of pentose phosphate pathway. In third stage, the conversion of 3 5-carbon sugars to 2 6-carbon sugars and 1 3-carbon sugars are catalyzed by 2 enzymes, transaldolase and transketolase. Transketolase catalyzes the transfer of 2 carbon unit from xylulose 5-phosphate to ribose 5-phosphate, yielding glyceraldehyde phosphate and pseudohaptolase 7-phosphate. TPP, thymine pyrophosphate, is a cofactor of transketolase. Transaldolase catalyzes the transfer of 3 carbon unit from pseudohaptolase 7-phosphate to glyceraldehyde 3-phosphate, yielding erythrose 4-phosphate and fructose 6-phosphate. The second transketolase reaction yields glyceraldehyde phosphate and second fructose 6-phosphate.